Monsieur le recteur de l'Université de Genève, Monsieur le président de l'Académie, Mesdames, Messieurs, j'ai envie de dire chers amis, parce qu'il y a des têtes qu'on revoit tous les 2 ans à ce Colloque Wright, j'ouvre le 15e Colloque Wright pour la science. Il y a à peu près 30 ans, Dudley Wright avait commencé avec ces colloques qui sont, au fil des années, devenus un événement important de la vie culturelle, et je ne dirais pas scientifique, mais quasi-scientifique de la population genevoise. Et c'est avec un grand plaisir qu'on accueille un public toujours plus nombreux, anxieux de se mettre au courant des derniers travaux scientifiques et des dernières évolutions dans les domaines que nous choisissons tous les 2 ans en fonction de l'intérêt et de la diversité. Cette année, c'est un sujet relevant de la chimie que nous avons choisi. Pourquoi? Parce qu'on ne l'avait jamais traité, la chimie, dans les Colloque Wright, et puis parce que c'est une année importante du point de vue historique pour la branche scientifique de la chimie, et qu'en plus, nous avons à Genève des industries chimiques qui sont discrètes mais importantes et auxquelles on tient particulièrement dans des périodes économiques difficiles qui touchent le secteur de la finance. Au fil des années, ce colloque est devenu aussi cher et important pour l'Université de Genève, et c'est pour moi ici, au nom de la Fondation, l'occasion de remercier très solennellement et avec beaucoup d'insistance l'Université de Genève pour l'intérêt, je dirais presque l'affection qu'elle porte au Colloque Wright pour la science et l'entrain qu'elle met, l'énergie qu'elle déploie pour aider à mettre en place l'organisation. C'est vraiment l'occasion pour moi de remercier les cadres de l'université, le rectorat en particulier, mais tous les professeurs qui se sont succédés pour organiser l'aspect scientifique des colloques, mais remercier aussi tous les gens qui s'engagent. Il y a des cellules comme la passerelle Science-Cité, le service de presse de l'université qui s'engage très fort, et beaucoup d'autres gens, les techniciens qui sont ici, les gens qui préparent la salle. Et vraiment, j'aimerais leur adresser mes remerciements les plus chaleureux. J'aimerais aussi saisir l'occasion, sans insister trop, parce qu'il ne faut pas être trop long, pour remercier les médias. Les supports médiatiques sont essentiels à permettre au but des colloques Wright à être atteint. Et nous sommes reconnaissants aux médias des articles qu'ils publient sur les conférences, les conférenciers et les sujets. Les sujets sont souvent ardus, mais les journalistes se donnent la peine d'essayer de les comprendre et de les retranscrire. Et c'est un soutien qui nous est essentiel. Et enfin, naturellement, il faut remercier très, très chaleureusement les conférenciers. En acceptant de venir à nos invitations, vous donnez à ces colloques, par votre notoriété, vos qualités d'orateur, le reflet, la brillance qu'il leur faut pour que l'université ait plaisir à accueillir les colloques dans son sein et pour que le public genevois soit impressionné par la qualité des messages qui leur sont transmis. Il y a un peu plus de 30 ans, Dudley Wright a fait œuvre d'un véritable visionnaire. Et comme me le disait le professeur Holmes yesterday, he was certainly ahead of his time. Il n'y a aucun doute, depuis lors les choses ont évolué, les scientifiques, la population et le monde politique a compris l'intérêt et l'importance qu'il y avait à ce que les sujets scientifiques les plus pointus soient plus ou moins compris par la population en général. Et je pense que les Colloque Wright, en ce qui concerne notre communauté, y ont largement contribué et ont peut-être fait, avec les votations auxquelles la population suisse a été amenée de participer, que la population suisse est probablement sensiblement plus au courant, consciente des enjeux des travaux scientifiques qui sont en cours actuellement. Mais si Dudley Wright avait cette vision, ce n'était pas simplement d'une façon purement altruiste et désintéressée. Je ne parle pas de son intérêt personnel, mais désintéressé pour la société. Les développements scientifiques dont il va être question, ou dont il est question d'une façon générale, sont intéressants pour la société parce qu'ils débouchent forcément un jour ou l'autre, tôt ou tard, sur des applications technologiques qui permettent des développements industriels et qui soutiennent l'économie. Et je pense que cet aspect d'application qui n'a rien à voir avec les conférenciers qui vont nous parler, qui eux sont des chercheurs purs de sciences fondamentales, mais cette application doit nous interpeller tous, en particulier la communauté de l'arc lémanique, qui a un besoin impératif de diversifier ses activités économiques de manière à mieux équilibrer le fonctionnement de cette économie locale. Je parle à dessein de l'arc lémanique parce que le canton de Genève est forcément largement dépassé, il faut voir nettement au-delà, mais l'arc lémanique est particulièrement bien placé pour se lancer dans ces applications avec des industries qui existent ou avec des industries nouvelles qui se constitueraient et qui se constituent, on lit ça dans les journaux tous les jours, et que ça, c'est un avenir extrêmement réjouissant et porteur pour l'ensemble de notre société locale. Je vous souhaite un très très bon colloque et je donne la parole au recteur de l'université qui va vous adresser quelques mots.
Madame Wright, Monsieur le Président de la Fondation, Messieurs les orateurs de ce colloque, Mesdames et Messieurs, ce soir, c'est au nom de l'université et en mon nom personnel de la reconnaissance que je souhaite exprimer, de la reconnaissance tout aussi chaleureuse et solennelle que celle que, Monsieur le Président, vous avez exprimée vis-à-vis de l'université. En effet, vous l'avez rappelé, nous ouvrons ce soir le 15e colloque Wright pour la science. Ça fait donc 28 ans que la Fondation offre à Genève, tous les 2 ans, une exceptionnelle semaine de partage entre la science et la cité. 28 ans que la Fondation soutient de fait l'une des 3 missions de l'université. Ces missions, vous le savez, c'est la recherche fondamentale, c'est l'enseignement, mais c'est aussi ce qu'on appelle les services à la cité. Et c'est là une notion un peu floue, pas tellement un peu diffuse, mais qui recouvre aussi bien le transfert de technologie que la formation continue, que très précisément le type de communication et de conférences que ce que dont nous allons bénéficier cette semaine. Et au fond, ce qu'on peut constater, c'est que la recherche universitaire, elle permet à la fois d'élaborer du savoir, des nouvelles connaissances. Par l'enseignement, elle peut transmettre ces connaissances, mais par ce service à la cité, elle peut également diffuser ces connaissances au sein de la cité, à tous les milieux de la cité. Je crois que ceci est extrêmement important parce que cette troisième mission de l'université, elle est à la fois fondamentale et elle est nécessaire. Elle permet de décloisonner l'université, mais elle l'insère également étroitement dans la vie de la cité et fait voler en éclats l'image de cette tour d'ivoire si jamais celle-ci a existé. Et je crois que, vous l'avez rappelé, Monsieur le Président, c'est ce que Dudley Wright a compris bien avant d'autres et c'est ce que la Fondation a su promouvoir depuis bientôt 3 décennies. Et la semaine que nous allons vivre, à cet égard et à d'autres, est exemplaire, puisque la Fondation nous offre 5 orateurs de la plus grande qualité, 5 conférences extraordinaires, et le panel de scientifiques qui vont nous parler cette semaine est illustratif de ce qui est une des grandes caractéristiques de la Fondation Wright, c'est d'associer non seulement la science la plus en pointe, la plus intéressante, mais une science qui est présentée par ceux qui la font et qui la font au plus haut niveau à l'échelle mondiale. Monsieur le Président, merci encore à la Fondation et à vous-même de nous offrir ces moments de partage et bonne semaine à tous. Alors, je passe maintenant la parole au professeur Alan Wright, organisateur scientifique. Alan Williams. Alan, tu ne m'en voudras pas. Je suis un peu perturbé parce qu'il y a un objet qui vient de tomber et d'éclater à mes pieds. Je vais reconstituer pendant que tu viens ici. Donc Alan Williams est l'organisateur scientifique du colloque. Il est également professeur à la section de chimie et biochimie de la faculté des sciences. Je crois que c'est un des individus qui porte la chimie dans notre université. Eh bien, il va porter avec nous ce colloque cette semaine. Merci Alan. Pendant ce temps, je vais essayer de reconstituer ceci.
Mesdames et messieurs, Monsieur le Président, Monsieur le Recteur, je voulais juste vous donner quelques indications pratiques pour ce colloque, ainsi que de vous esquisser brièvement le programme de la semaine. Vous aurez remarqué que les conférences seront données en anglais cette année toutes, donc un petit aperçu survol en français, quoique avec un accent anglais, vous serez peut-être un peu utile. Du point de vue pratique, la conférence sera donnée dans quelques instants, si je ne parle pas trop long, et ensuite, ceux qui veulent partir sont priés de partir tranquillement et assez rapidement. Ceux qui veulent poser des questions sont évidemment plus que bienvenus pour rester. Vous aurez reçu à l'entrée des petites fiches sur lesquelles vous pouvez marquer des questions qui peuvent être assez générales. Ils seront ramassés par des hôtesses et très rapidement après la fin de la conférence, nous espérons ouvrir le tableau ronde où tous les conférenciers s'exprimeront sur les questions posées. Donc je vous inviterai chaleureusement à poser des questions. Maintenant, il me paraît utile de vous dire quelques mots sur le sujet et thème de cette semaine, architecture moléculaire. Ce n'est pas vraiment la chimie, c'est l'architecture et moléculaire. Architecture, ça veut dire quoi? C'est une science de la construction, mais qui importe deux choses aussi. D'une part, la fonctionnalité. Il faut que ça sert à quelque chose. Un bâtiment qui n'est pas fonctionnel n'est pas trop apprécié. Et aussi la beauté. Alors, le mot moléculaire est un peu utilisé comme une étiquette. Maintenant, nous avons eu la biologie moléculaire, nous avons la gastronomie moléculaire, nous aurons bientôt les impôts moléculaires. Mais dans ce contexte, je voulais dire, ça donne l'échelle. C'est des objets qui sont construits à partir des atomes. Et l'architecture moléculaire s'exprime dans tous les matériaux qui nous entourent maintenant et jusque dans les processus vitaux de notre propre corps. Donc, c'est seulement par hasard, 100 ans presque au jour, que les premières images des atomes dans l'espace ont été obtenues en 1912, d'abord par Max Lowe et ensuite par père et fils Bragg. 50 ans plus tard, c'était l'année où 2 prix Nobel ont été donnés à Watson et Crick pour la structure de l'ADN et puis à Perret et Kenru pour les premières structures des protéines. Donc il y a une évolution sur 100 ans. Nous ne parlerons pas de l'histoire dans ce colloque, mais plutôt des résultats tout récents et aussi, j'espère, de l'avenir, parce que nous souhaitons faire ressortir les questions qui doivent être résolues à l'avenir. Et donc, il y a 2 aspects. Il y a d'une part, ces 10 dernières années, nous avons pu obtenir les structures des molécules de plus en plus compliquées, contenant peut-être des centaines de milliers d'atomes. Et d'autre part, le côté créatif du chimiste de synthèse a pu construire des matériaux nouveaux avec des propriétés étonnantes. Et le colloque essaie de montrer les deux choses. Et ils ne sont pas séparés, parce que l'observation de la nature inspire la créativité du chimiste de synthèse. Alors, très brièvement, nous commencerons aujourd'hui avec le canal de potassium présenté par Ulrich McKinnon. C'est un exemple classique d'une structure qui est construite, fonctionnelle et belle. C'est une petite contrôle d'une petite fonction, mais c'est une petite fonction importante quand vous savez que parmi nous tous, 25% de notre énergie actuelle est utilisée pour transporter le sodium et le potassium à travers les membranes cellulaires. Demain, nous irons dans le monde des matériaux. Nous allons voir une approche novatrice vers la création des matériaux avec le professeur Takuzo Aida, des matériaux qui sont construits essentiellement à partir de l'eau. Bon, ce n'est pas totalement nouveau parce que je vous révèle que je suis moi-même un matériel construit essentiellement à partir de l'eau, étant à 70% de l'eau, mais je suis sûr que les matériaux du professeur Aida sont bien plus performants que moi. Ensuite, mercredi, nous entendrons le monde des polymères. Polymères qui est un petit peu plus que le sac en plastique que vous prenez au supermarché, mais qui ont des propriétés électroniques remarquables, permettant d'une part d'émettre la lumière et d'autre part de l'absorber et transformer en électricité. Et puis, jeudi, nous toucherons un domaine qui commence seulement à évoluer, la construction des machines à l'échelle nanométrique, à l'échelle moléculaire. Et il faut savoir que la manière classique de construire des choses, c'est que le sculpteur, il prend un morceau de marbre, il le fait toujours plus en plus petite. Les machines dont vous parlera le professeur Lee sont construites à partir des atomes que vous mettez ensemble de manière à ce qu'ensuite ils peuvent bouger et bouger autre chose. Et enfin, nous allons terminer avec la chimie industrielle, carrément avec une usine. C'est une usine que nous avons dans chacun de nos cellules qui construit constamment les protéines qui sont en train de faire pousser vos ongles, faire pousser vos cheveux, plus ou moins selon les cas, et qui permet de traduire de manière sans faute l'information génétique en protéines que nous utilisons pour toute la vie. Donc, je vous souhaite très vivement de passer une semaine, d'émerveillement avec cette science au plus haut niveau. Je réitère les remerciements de M. Maître Patry aux conférenciers. Il faut savoir, tous sont des scientifiques actifs qui ont bien voulu quitter leur laboratoire pendant une semaine pour venir expliquer au grand public genevois ce qu'ils sont en train de faire. Je dois juste malheureusement vous prévenir que nous avons eu un petit problème audiovisuel aujourd'hui. Monsieur McKinnon a un magnifique Macintosh qui est un poil plus avancé pour notre système audiovisuel et le résultat, vous verrez bien les choses, mais il y a une distorsion un peu malheureuse que les images vont paraître quelque peu élargies par rapport à leur hauteur. Donc, ils ne seront pas tout à fait aussi beaux qu'on aurait souhaité. J'ai rencontré le même problème avec la glace à la maison, parce que des fois, ça paraît un peu plus élargi par rapport à l'auteur, mais pour le moment, on n'a pas encore découvert de remède. Ceci dit, je suis sûr que vous aurez beaucoup de plaisir à écouter cette conférence, et je donne la parole à mon collègue Howard Ritzman, qui est directeur du NCCR en Chemical Biology, qui va présenter le conférencier. Merci beaucoup.
So it's a great honor to be able to introduce our speaker today. So for those of you who want to change your headphones, you won't have to écouter mon misérable accent en français. A crucial step in the evolution of life on Earth is the creation of a cellular membrane. And the cellular membrane allows the cell to compartmentalize its functions and also allows cells to communicate with other cells and control their internal environments. The cell membrane acts as a barrier to the outside and selectively allows certain molecules to pass through in a controlled way. And it also allows hormonal interactions between cells and communication. And during the evolution of life, it allowed multicellularity and the development of life as we know it today. Now, one of the important molecules that crosses these cellular membranes are ions. And our topic today is the transport of these ions across the cellular membrane, in particular potassium. Very important in all tissues, but especially in the brain. So our speaker today, who is one of the most distinguished people in the world in this field, of course, started science very early. In fact, at 11 or 12 years old, he spent a summer in a science enrichment course, and I think this had a very strong effect on his future interest in science. He then obtained a bachelor's degree in biochemistry from Brandeis University, an MD at Tufts University Medical School, and actually performed a medical residency, and I guess was on his way to become a medical doctor, which I guess he is since he completed his residency, but then decided to concentrate and focus on basic science. And I think this is a great thing for us because we've learned a tremendous amount from his work, his basic research. And he went to do, after his medical training, a postdoctoral work at Brandeis University. And in 1996, he took a position at Rockefeller University, where he's been ever since, and is now a Howard Hughes medical investigator. He's received many prizes. I'm not going to list all of them for you. I'd just like to say that in 1999, he got the Albert Lasker Award. The Lasker Award is frequently given to people who later on get the Nobel Prize. And in this case, it happened again. And in 2003, he received 2 other prizes, with some others in between. He received the Louisa Gross Horwitz Prize and the Nobel Prize in Chemistry. So I'm very happy now to pass the place on to Roderick McKinnon. We're very glad that you could be here with us.
Thank you very much. I'm very happy to be here, and I'll do my best to explain the subject that I work on in science, which is the electrical system of life. I'll talk about the molecular architecture of a class of molecules that are responsible for the electrical system of life. Before I start to talk about the molecular architecture, though, I want to talk just a little bit and introduce this subject of the electrical system. And the reason is, I think you all know that living cells, that living organisms make electrical impulses because you are aware of electrocardiograms and electroencephalograms, which measure electrical signals from the heart in the brain. And you know that nervous impulses allow us to move. For example, I think to wiggle my finger, and I think you know there's an electrical impulse that travels down from your brain through the spinal cord and out to the muscles in your hand. And so, although we're all aware of this, at the same time, it's a little strange because we also are aware of most electrical devices like computers and iPhones. They are electrical devices and they have no water in them. And in fact, we know if we put them in water, they don't work. And yet we also know that we're mostly water. So how come our electrical system works? So I want to say a little bit about that before I talk about the molecular architecture. So the idea of electrical signals in living things was started several hundred years ago, the late 1700s, by an Italian physiologist named Luigi Galvani. And he did experiments where he first discovered that if he took a metallic arc here, and he took a freshly prepared frog legs and nerves going down, and he touched here and here, there was a contraction. And he interpreted it. Now, at the time, there was not a lot known about electricity. Even the physics of electricity was very limited. People knew about static electricity. That's the kind, on the dry winter day, you walk on the rug, you touch the doorknob, you get a spark. A little more sophisticated than that, people made electrostatic generators, and there was a kind of device that could store this static electricity called a Leyden jar, but not much was known. But Galvani knew about that work, and he made the analogy in his mind that there must be something electrical to make the muscle contract. Now, I'll just make one little mention about a side story, and that was another scientist, a physicist named Volta, looked carefully at Galvani's experiments and wondered why Galvani used a piece of metal that was made of two metals, zinc and copper. And Galvani never explained that to anybody because he didn't know. Volta focused on these two pieces of metal, and in fact, he said, it was a very big controversy, he said there is no animal electricity because it's metal electricity, and it had to do with putting two pieces of metal together. And in fact, Volta worked on this very hard, was a very smart man, and he took discs like coins of zinc and copper, and stacked them with gauze material in between with salt water in it, brine. And he basically invented the first battery. And he showed that this stack of different metals would make an electric current if you connected the two ends with a wire. And what's interesting about that, so Volta was right. There was electricity in the metal. And in fact, what's interesting is because he could make with his battery the first sustained electrical current in a wire, it was shortly later discovered that there was a magnetic field around that wire. And a scientist named Ampere looked at that magnetic field very carefully and came up with a quantitative relationship between that magnetic field and the electrical current. And in fact, then a few other scientists, one named Faraday, and also said, well, wait, if an electric current makes a magnetic field, then maybe a magnetic field can make an electrical current. And that was true. So if you move a magnetic field, spin a magnet in a conducting wire, you can make a current in the wire. And you'll recognize that as an electric motor. So then, and that was not too many years after Galvani's experiments. And shortly after that, a physicist named James Clerk Maxwell, having all the data from Faraday about electric currents and magnetic fields, came up with his theory of electricity and magnetism that actually took two totally different forces and combined them into a single unifying theory that in fact connected light to those forces. So the reason I tell you that is because it was actually this physiologist who made this observation that made Volta try to disprove him and did experiments that in less than 50 years led to the theory of electricity and magnetism. So it's an interesting thing how one science affects another science. But back to the electricity in living things, it turns out Galvani was right. The electricity that came out of this metal arc wasn't actually responsible. It was initiating the contraction, but the contraction itself had everything to do with electricity that was spontaneously arisen inside the living tissue. And in fact, it's taken us about 200 years to figure out what's going on by work of many different scientists. But if you take, in this simple hand-drawn picture that I made, if you take any living cell and you take a voltmeter and put an electrode inside the cell, here's the cell, and you put an electrode inside the cell and the ground wire outside the cell, the voltmeter will read a voltage, typically between minus 50 millivolts to minus 100 millivolts. So a millivolt is 1,000th of a volt. So 100 millivolts being 1/10 of a volt. Not a big voltage, but actually a substantial voltage. All living cells have it. And you can see that I have some symbols here for sodium and potassium, and I'll explain why I'm showing that. How does this how does this work? So in other words, what I'm describing is that the membrane of all cells, all of our cells, is electrified. And so to understand how that happens, let's take a little piece. Imagine we're looking at I'll go back. We're looking at this picture, and imagine we now take we take a piece of the cell membrane, and we could look at it. So this is a cartoon of what we might see, or something like what we might see. You would see that there is a membrane made of lipid molecules that form a double layer called a bilayer. This would be outside the cell, and this would be inside the cell. And inside that membrane, embedded in the membrane, you would see proteins that live inserted inside the membrane. There are many different kinds of proteins in the membrane, but there are certain ones that transport ions. And of the ones that transport ions, there are two major classes. One class are called the pumps. They're called ion pumps. And the other class are called ion channels. Now, the difference between them is this. An ion pump uses energy from ATP hydrolysis. So this is a high-energy molecule inside the cell that is converted to ADP plus phosphate, and the energy of that conversion is captured. It's like the petrol in an automobile to make it run. The running of this pump pumps sodium out of the cell and potassium into the cell. So this pump continuously works, pumping sodium out, potassium in. And what happens when those work for a while on a cell is you have high sodium on the outside, shown as red circles, and high potassium, shown as green, on the inside. So this pump uses the chemical energy to make a gradient of ions. And the gradient is high potassium inside, high sodium outside. Now the channels are simpler than the pumps, because when a channel is open, the ion goes through it, but it goes down its gradient. So if a potassium channel opens up, because potassium is at a high concentration on the inside, it starts to flow out. Just like, if you inflate a tire of a car and you punch a hole in it, the air's going to go out because the pressure's higher on the inside than the outside. In this case, there's more potassium in than out, so open a potassium channel in this membrane and potassium goes out. Now the ions, potassium and sodium, when they're in water, carry a charge. So salt in the kitchen, sodium chloride, put it in water. It dissociates into sodium with a plus, chloride with a minus. Potassium chloride, same thing. So potassium is carrying a positive charge, and when potassium goes out, it's carrying a charge out. And what that does is it leaves negative charge on the inside and puts positive charge on the outside. So it's because potassium flowing out carries charge, it electrifies the membrane. And that is why all of our cells have a charge across the membrane. Now imagine if the potassium channel closed and a sodium channel opened. So now the sodium could go in. Sodium goes in, the polarity will flip. So it will become positive inside and negative outside. in simple words, this is the origin of electricity in living things. It's because the cell membrane is an electrical capacitor, and the pumps make gradients, and the channels dissipate those gradients to charge the membrane. Here are some examples of what a few different kinds of channels do for us. I just talked about how potassium channels charge the membrane. A channel that conducts calcium, so a special one that recognizes calcium, if it opens, calcium, which is normally low on the inside of the cell, enters, and the calcium, depending on the cell, will do something like cause muscle contraction. So when calcium channels open in a muscle cell, the calcium ion causes the muscle filaments to contract. In some cells, some neurons, when the calcium enters, it actually affects some enzymes that lead to turning on of certain genes. So in this case, we call the calcium channel, letting calcium go in, acts as a second messenger to make another process happen. In the kidney, in some cells, we have potassium and chloride channels, and when the potassium and chloride channels are both open, potassium and chloride move, water follows, and this is how electrolytes in waters are secreted. And so this is just a few of the examples of how these channels work in physiology. Now, you don't have to remember all the details. I want to say two concepts. If you think about these channels and how they work, there are two requirements. One is they have to be selective for a particular ion. So, for example, when a potassium channel opens, if it led the sodium ion in and the potassium out, the two positives would go against each other and nothing would happen. So it has to select for potassium. And the other thing is, the channels are not open all the time. They have to open at the right time. So there has to be a way to open and close them. It's called gating. And they have to respond to the right signal so the cell can make it open at the right time. Now, what I want to talk about now is the molecular architecture of how some of these processes happen. How does a potassium channel let only potassium through? And how do potassium channels open and close in response to specific stimuli? Just before I talk about that, I want to show you just briefly how we determine the molecular architecture. In our case, we use a technique called X-ray crystallography. And in this technique, you have to first make a crystal of your protein. And in the case of proteins that are in cell membranes, you have to, so imagine here is a little picture of a cell membrane with proteins in it. You have to take the proteins out of the membrane, and for that we use detergents, which are lipid-like, or we use mixtures of detergents and lipids that don't quite form a membrane, but put a little coat on the membrane proteins, in our case, ion channels. We purify the ion channels, and then we find conditions under which the channel produces a crystal. Here's a picture of a protein crystal of an ion channel. This bar is 50 microns, so it's 50 times 10 to the minus 6th meters. It's rather small, but that has a lot of molecules of ion channel in it. And there's something interesting I want to tell you about protein crystals. People often think a protein crystal must be something very unnatural for a protein. And so how would we know that when we look at a protein in a crystal, that it's the same as a protein in the membrane, for example? And that's something you have to be careful and test. But in almost all cases, it is. And these crystals are not like sodium chloride crystals. They are very precise arrangements of the protein in a regular array, but they have a lot of water in them. A typical protein crystal is 60 to 80% water. And in fact, if you crystallize an enzyme and then put the substrate for the enzyme, that is the molecule that it will do the reaction on, that reaction will work perfectly. In the case of ion channel crystals, you can put the ion in that the channel selectively binds, and they will bind in the ion channel. And the ones that do not bind will not. So the crystal is actually a rather natural condition. And in fact, it's why it's very hard to grow these crystals. You have to find conditions under which the protein will tolerate being taken out of the membrane and will form a crystal. So once you have the crystal, you put it in an X-ray beam. Usually for our crystals, we put them in an X-ray beam at a synchrotron, a very bright source of X-rays, and you get a pattern that looks like this. And of course, there are a lot of little spots on here, and this doesn't look like anything currently. But it's with this information, and in fact from this information from different orientations of the crystal, that you get the data, which you can't immediately turn into a picture of the molecule. There has to be additional information in it, and the information is this. This gives us the equivalent of a completely unfocused image. And to get the focus on the image, you have to do some additional experiments, and once you have that additional data on a computer, you can focus it. And then you get a picture that looks like this. This is blue. The blue mesh is called electron density, and you can see it has a coherent shape. It starts to look like something, and in fact, you can recognize amino acids, that is, the building blocks of a protein, and you can build your molecular model into this electron density, and then you can find the optimal model that best matches the data, and then you have determined your structure. Let me make a comment about the way we represent proteins. If you look in a book with proteins, you'll see things that look like this. What I'm showing here are 4 representations of the same protein. As you'll see, it's part of a potassium channel. This is called a wire representation. This is called a ribbon. This is called a stick. And here, in this case, the atoms all the atom positions are shown, connected by bonds. And in this case is an all-atom, more realistic atom size of the protein. Now, the reason I show you this is because proteins really look like this. They're very, very condensed structures. There's not much space between the atoms. They're packed together. They're very incompressible, like water. You can't squeeze them. They fill up the space. But I will often show representations like this, because in a big protein structure, if I show you this, it will look like fish eggs, and you won't see anything. In this kind of representation, we just make a ribbon along the polypeptide chain. Proteins are built of building blocks linked together in a polymer out of 20 different amino acids, and they fold up to take these particular structures. And so when you look at a picture like this, just remember that actually this is a subterfuge or a simplification of something that really has many, many atoms and fills up the space. Okay. One other thing I want to show you. When we study these ion channels, we not only look at their structures. The subject of this colloquium is molecular architecture, but these proteins have function. And I just want to show you 2 ways that we look at the function of ion channels. And it's because when I show you some of the ion channels and the architectures, I want to correlate those with some function that I will show you. So this is called 2 ways to measure ion channel function. In the top, we've isolated a tiny patch of membrane from the cell with 1 ion channel in it. And this is a particular ion channel that the name isn't important right now. It's called a voltage-dependent channel, and I'll show you that near the end of the talk. But the important point is, here we can change with an external amplifier. We can change the voltage on the membrane and look at the channel as a function of the membrane voltage. In this particular channel, as is shown here, at minus 100 millivolt membrane voltage, the channel is not open. But then when you step the membrane, to plus 100, you see that the channels sometimes open. And when it opens, so this is current, measured here, and this is time. So what's happening is the channel is opening, boom, and current runs. How much current? This is 20 picoamps. So that's about 10 picoamps of current. A picoamp, that's 10 to the minus 12th amps. So typically in the house, we have amperes. But we're talking about small currents. But how small or big a current is actually depends on the circuit it's in. And in this circuit, this is a reasonably large current. Has to do with what the capacitance of the cell membrane is. But the important thing is, to understand is when this channel opens, you see the current jump up, and it's at a different level. So that would be like if this were a faucet turning on and off, this could be water flow, and here would be zero water flow, and here would be some amount of water flow. And what you're watching here is the faucet turning on and off. It comes on, and it goes off, and on, and off, and on. It's jumping back and forth. And that's an interesting concept that if I should mention that when you study molecules and things small, in the architecture of things small, it's a little different than talking about the architecture of a building. And it's different in the following way. A building is big and heavy. And when we look at it, it sits there. And it keeps its form. But when things are very small and not very heavy, they're subject to thermal motion. So if we looked at a protein in real time, if I'm a building, here I am, a cathedral. But if I'm a protein, I'm getting bumped around all the time. And this does not have to do with the fact that, proteins are shaky compared to buildings. It actually has to do with the fact that a building is very large. The building also is feeling the thermal fluctuations, but the atoms hitting the building are not strong enough to make it shake perceptibly. But once something is sufficiently small so that it's undergoing molecular collisions, it vibrates, it fluctuates. In fact, the whole world of biochemistry and the way we work in our brains and when we think, although the signals you'd think are smooth and precise, they're called stochastic, because everything is subject to thermal fluctuations. And in fact, it's one of the incredible things about life, that life can give rise to organisms such as ourselves who can do very sophisticated things like talk and listen and interpret. And this is all happening in an architecture that's always undergoing thermal fluctuations. So here you see what's happening is we would say, as people who study molecules, we would say, the probability that this channel is open at minus 100 millivolts is very close to zero. But at plus 100 millivolts, there is definitely a finite probability that this channel will be open. Now that's a tiny patch of membrane, isolated, so we're watching one molecule open and close. But if you now look at the whole cell where these channels are operating, we do the same change in voltage. We see a big current turn on, and then when we step back, it goes inward, and then it shuts off. And this is the same as this, but what happens is there are thousands of these being averaged opening here, you see? And so underneath this big smooth signal, in fact, it's not completely smooth. You can see it's jiggling a little bit, and it's jiggling a little bit because actually there are fluctuations of thousands of them happening. But this is the current due to a few thousand of these. And in fact, if you look at this is 20 picoamps, and that's 10 nanoamps. And a nanoamp is 10 to the minus 9th amp. So this is, in fact, 1,000 times bigger than that. So there's a much bigger current here due to many, many channels operating. Okay. There are two different ways of looking at the same thing. Now, potassium channels. So how many potassium channels do we have in our bodies? 78 is my last count. 78 different genes that encode potassium channels. And what this is called a family tree or a dendrogram. And so if you follow, the end of one of these would represent a channel gene. And if you count around, this thing has 78 little branches on it. And these are like branches of a tree. So you can see this little cluster here. They're all related to each other. And they're a little less related to this. And they're less related to these. And much less related to these. Totally different branches. So we call these clades or families within the big potassium channel family. There are 5 different major families of potassium channels. How come so many? How come 5 families? How come 78 different ones? Well, it turns out different cells need different potassium channels to respond to different signals. And that's why they're different. And I will show you a few examples. But first, why are these potassium? What makes these potassium channels? I tell you that there are many different ones and they're very different, but they must have something the same. And they do. The part shown in red is very similar in all of them. And the 2 linked together here have a simple explanation. And what they have in common, so we'll look at the part of this family tree of potassium channels, the architecture that is the same. And you might guess that the part that is the same is the very part that makes it a potassium channel. So probably potassium channels all evolved from a common ancestor, potassium channel, and then evolution led to the diversity of different kinds, but they all have a structure that looks like this. So what I'm showing you here in this rotating movie are the 4 subunits of a potassium channel that span the membrane. So the membrane would run from about here to here. So outside the cell would be up here, and inside the cell would be here, and the membrane is horizontally disposed on the sides of this thing. And it makes a pore across. So you see 4 identical subunits. It has 4-fold symmetry, and it makes the ion pathway. Here's a simpler representation. I'll take away 2 of the subunits, and we'll just look at 2 across from each other. And you can see down the middle the ion conduction pore. And so remember what I told you, that actually proteins have atoms that fill the space, and these are simple representations that are just following the polypeptide chain, so that we can just get a feel for the shape of this thing. But what's interesting is inside here I show some electron density with some atoms in it. This seems relevant because these are potassium ions inside the protein, and in fact that's a potassium ion surrounded by water molecules. You can see the electron density for this. And so when you look at this, if I ask anyone in the audience, which part of this looks like it makes it the potassium channel, I know you would all get it right. You would say it must be this part that is directly interacting with these lined up potassium ions. And in fact, they're potassium ions that are don't have water around them. And so let's look at, it turns, this part shown in yellow here, if you look at the genes for potassium channels, from organisms ranging from bacteria all the way up to human, across plants, the part that was shown in yellow is almost the same in all of them. And by the same, what I'm showing here in blue are what's called the single letter code for the amino acids. So amino acids, as I mentioned, are if a protein is made of beads on a string that fold up to take a shape, the amino acids are each bead. And there are 20 different types of beads. And scientists, for shorthand, have associated each of those 20 with a one-letter name. TATTVGYG, that stands for threonine, alanine, threonine, valine, glycine, tyrosine, glycine. And if the point I want to make here is that all through the tree of life, that's almost the same. And that's what tells you, aha, right from the gene level, you can tell however the potassium channel is selecting ions, the secret must be held in the structure of this thing that is almost invariant. And in fact, it's why I wanted to become an X-ray crystallographer, to see this. And it looks like this. So here's the detail of that strand that I showed in gold or yellow color. And now I'm showing it as a stick representation, which is at each little point, each little bend in this is an atom. So the ends of these are atoms. In important atoms of the protein, I've shown as red spheres. Those are oxygen atoms from the protein backbone. And they're all pointing in toward the center at the potassium ion, shown in green. And so what you actually see is that a potassium ion, each one, is sitting in a little cage with 8 oxygen atoms around it. 4 on the top, 4 on the bottom, for each of these cages. Now, what does a potassium channel have to do? Remember, the first important thing is it has to conduct potassium and not sodium. And potassium and sodium, when they're in water, are surrounded by water molecules. And in fact, the oxygens from the water molecules point at the plus charged metal, or plus charged potassium. And what this protein has done is its structure has made a mimic of the water hydration shell, so that the potassium ion can, without a large energetic cost, come out of the water and be bound in a cage by the protein. And it turns out sodium, interestingly, is smaller, but it doesn't go in there. And in fact, experimentally, if you try to put sodium in there by taking the potassium away, the structure changes completely. It collapses in the center, and no ions bind at the center. This structure is only stable with potassium in it. What a beautiful way. Imagine if you were around at the beginning of time and were asked, hey, we need a potassium channel. Can you make a potassium channel? How would you do it? Pretty good if you came up with a protein structure that could only maintain its conductive structure if it had potassium in it. And so what you're seeing here is that the protein is a mimic of the shell of waters for a potassium ion and not well enough for a sodium ion. Turns out the sodium costs more energetically to dehydrate, and you don't get the same energy back when you try to put it in here because it doesn't contact all 8 of those oxygens as well. And so what you have is this structure that is very well tuned in size for binding a potassium ion by mimicking the water shell on the potassium ion in their sequential binding sites, so the ions can come in and diffuse through from outside to in or inside to out, depending on the gradient that is set up by the pump. Remember, there's no pumping here. It's just a channel that lets the ion go, but it only lets the right kind of ion go. So that's what makes these potassium. So you can see it's the architecture and chemistry of this part we call the selectivity filter that is shared by all of the potassium ions in nature that we know of. So far, no exceptions. Next, I want to address the other question I raised earlier, and that is, how do they open and close? How do they get turned on and off? And in that case, what I'm going to do is give you 3 examples out of this 78, and we certainly don't know them all. We sort of know the structure of one, essentially, at least one from each of these. It's most of one from here. But I'm going to take 3 examples that we're starting to best understand at this point and show you how we're beginning to understand how the cell tells the channels to open and close. So the first case, example 1, is a potassium channel that is regulated by what's called a signaling lipid. Now, this kind of channel is actually the kind of potassium channel that does the job that I introduced in the very beginning. It is a channel that controls the resting potential. So you have the potassium-sodium gradient. It sits open in the cell membrane and makes it negative inside by letting potassium go out. And some cells, though, can control the level, how many of those are open. And they do it. They do it by having this channel respond to a specific lipid. So the cell membrane is made of lipid molecules, but there are some special lipid molecules in PIP2 phosphatidylinositol 4,5-bisphosphate is one such. When it binds to the channel, it causes it to open. Now, PIP2 level in the membrane is controlled by many, many different enzymes. There are enzymes that can pull a phosphate off, or enzymes that can add a phosphate, enzymes that can convert PIP2 into other molecules by cutting the whole inositol phosphate head group off of it. So this is, in a sense, like it's a molecule in the cell membrane that is controlling this channel, and the cell has an ability to make a lot of it or a little of it to regulate many processes, including the activity of this channel. So let's look at how PIP2, how would you tell how, let me just show you an example of what happens if you look at a membrane without PIP2 in the membrane or with, and this is a membrane with one channel. Here is a case with no PIP2, and again, this is a single channel representation. So what you're looking at is current in time. This is no current, C for closed. There's no channel activity here. This is 1% PIP2 in the membrane. Here is closed and here is open. What you see in this membrane compared to that membrane is this one channel is open almost all the time. Here it closed and then it opens. In fact, it does close very briefly. You see them as little brief spikes, but it's mostly conducting when the membrane has the lipid composition of the membrane is 1% PIP2. So how do we understand the architecture of how it's working? Well, easy. Determine the structure of this potassium channel in the presence and in the absence of PIP2. When we do that, we see the following. So this will start to rotate in about 3 seconds. So this is the channel without PIP2 in the crystal, and this is the channel with PIP2. And in fact, PIP2 is bound here, one on each subunit, so there are 4 in a ring. And you notice there's an architectural difference here. This channel spans the membrane, and this unit attached to the channel extends into the cell, into what's the cytoplasm of the cell. Notice that it's moved up tightly against the transmembrane part. And it's because PIP2, in a sense, has made interactions to cause that to happen. So in the absence of PIP2, it looks like this. The presence, it looks like this. So now I want to show you a movie, what's called a morph movie, between this structure and this structure. And realize when I show you this movie, the endpoints of the movies, that's based on the data. Those are crystal structures shown here and here. The in-between is actually using Hollywood techniques, essentially, to watch the transformation in between. Okay. But it gives you a good impression of what has to happen. So what I mean is we don't have all the structures of all the intermediates, but we can imagine something like what they must look like. So here is the channel starting out in the absence of PIP2, and the PIP2s, they're poised to come in. And notice when they do, there's a movement up. Oops, sorry. Let's start again. The pointer has many buttons, and I keep hitting the wrong one. And now I can't find my pointer here. Oh, there it is. PIP2 binds, and this comes up. So now we'll zoom in and look closely. When the PIP2 binds, the phosphates make specific interactions with these amino acids called arginines, and it causes it to lift up. And notice these arginines here, they're positively charged. They're positively charged inside, and PIP2 causes them to come out. Now imagine this is a potassium channel where positive potassium ions flow through. It starts out with these positive charges along the ion conduction pore, which would make an electrostatic barrier. It would repel potassium from going through. The PIP2 binds, causes those to come out, associated with the conformational change. Also, the helices began to splay here and become a little wider. So I represent this here, that if we look at what happens is there's a conformational change caused by the binding of the PIP2, which causes this part of the structure to move up, open the helices a little, and move arginines out of the way. So you go from here to here, and arginines come out of the way. So in a sense, this is a very simple thing that's happening. The lipid molecule is binding, changing the shape of the protein, and opening the ion conduction pore. So there we can understand, based on the architecture, how that channel opens and closes. Example 2. Example 2 is a channel that we call GERK. It's a G protein gated channel, and I'll explain what that means. That's a channel that is in the membrane and will be activated by another protein called a G protein beta gamma subunit. The G protein beta gamma subunit is released from another membrane protein called a G protein coupled receptor. And in fact, the Nobel Prize in Chemistry this year was given to two scientists who figured out many of the mechanisms of how G protein coupled receptors work. In particular, one called the beta adrenergic receptor. There's a membrane protein, and it's bound to G protein alpha and beta gamma. When a neurotransmitter or a drug binds to the G protein coupled receptor, these are released, and in the case of a potassium channel, the beta gamma subunits bind and cause it to open. Now, this mechanism I'm going to show you has an interesting history. This mechanism controls your heart rate, this potassium channel. And the history is, in fact, there was a German physiologist named Otto Loewi, and Otto Loewi made the following discovery. He discovered that he knew that if you took a frog heart, isolated from the frog, and you isolated a nerve that goes to the heart called the vagus nerve and stimulated it, the heart rate slows down. So many people knew about that. And there were great debates about how does the nerve tell the heart to slow? There were two main ideas. One idea was that they were by now people knew about electricity. Not the details, but they knew about electrical impulses, and so they thought, one idea was that the electrical impulse goes directly to affect the electricity of the heart without any chemical intermediary. In other words, there was some electrical couple between the nerve and the heart muscle. The other idea was that maybe at the end of the nerve there was some transmitter, some chemical transmission, and there were some reasons for people to wonder about that. So Loewi actually did the experiment where he stimulated the heart, very overstimulated it. He collected the perfusate, and then he applied the perfusate, to another frog heart that had all the nerves peeled off, and it slowed down. This was very important because this was the discovery of chemical transmission or synaptic transmission. And in fact, Loewi, it turns out the transmitter was acetylcholine, the important neurotransmitter in the cholinergic nervous system. Acetylcholine had been discovered by Dale, and Loewi and Dale shared the Nobel Prize in the 1930s for the discovery of chemical transmission. So there's a little interesting side on Otto Loewi's experiment. It's hard to believe it's true, but this is the story. He said he dreamed this experiment one night. He said he was in bed and he was asleep and he had a dream about doing this experiment, and he woke up and he wrote it down and he went back to sleep. And then he said he woke up the next morning and he couldn't read it, couldn't read his handwriting. That's what he said. So then he said he was very frustrated, but on another night, he had the dream again. I find this hard to believe. And this time, when he woke up, he said, I'm afraid I won't read my handwriting. So he went straight into the lab in the middle of the night, and he did the experiment. And the heart rate slowed down, and he was very happy. Can you imagine saying, I had a dream, and I did an experiment that was so important, I won a Nobel Prize, and I wrote it down, but now I can't read my handwriting? But that's the story behind it. And in fact, you can, in our laboratory, we can do a little simplified version of this same experiment. But in this case, you take a frog egg, an oocyte, and you express by putting RNA encoding the proteins, and then the proteins are synthesized by a process that you'll hear about from Venki Ramakrishnan later in the week. But you express both a GPCR called the muscarinic acetylcholine receptor and the G protein gated channel in this cell. And now you record electrical current, and you can see this is not a single channel recording, but it's from a whole cell. So you recognize the big smooth current here. This is current and time. So you put some potassium chloride outside, and there are some potassium channels that are normally in this egg, and you can see a small potassium current. And then you add acetylcholine, and you see a big potassium current. And now you, just to make sure all this current was due to this channel, you add an inhibitor called tertiapin Q that you isolate from bee venom, and that inhibits this channel. And you see that all the acetylcholine activated channel is inhibitable. So what this really means is that all this current turned on was because you add acetylcholine, it stimulates this G protein coupled receptor, which releases its G proteins, and beta gamma somehow activates the G protein, activates the channel to open it. So the question is, how does it turn this current on? And so, you solve the structure in the presence of the G protein beta gamma. And if you do, you see I should say we know all the parts for this. And the parts are because we had solved the structure of the G protein channel. And in fact, it looks a lot like the other channel I showed you that is PIP2 activated. And in fact, PIP2 is indeed bound to this channel as well. Here is the G protein coupled receptor bound to alpha beta gamma that was featured in this year's Nobel Prize work of Brian Kabilka. And somehow the beta gamma subunits have to come over and turn this on. And so if you solve the structure of the potassium channel in the presence of the beta gamma, something very interesting showing here. Nature is very smart. Notice that this squiggle drawn here that's attached to these proteins. That squiggle is attached there because it's a lipid, covalently linked onto the protein, so that when this is released, it doesn't go away from the membrane. It just diffuses under the membrane. So it doesn't go floating off away from the membrane. It stays in the membrane, very clever, and diffuses, finds the ion channel, and binds to it. And what happens when it binds to the channel is it binds this way. So here is this G protein activated channel, the kind that's in our heart that controls the heart rate. So imagine the membrane leaflet. This is outside the cell, and from here is inside. So all of these things are inside, but just below the surface of the membrane. And in addition to PIP2 for this kind of channel, each subunit is attached with beta gamma. So this is a very big protein. If I showed you with all the atoms here, it would look like a meteorite. But this is what it looks like. Now, how does that open the channel? Okay, so what I'll show you here is a movie of this potassium channel without the G protein, and then what happens when the G protein binds. But I won't show the G protein because it will get in the way. I just want you to watch what happens to the structure, to the architecture, when the G protein binds. So here it starts unbound, and when the G protein binds, it twists. Oops, I did it again. Okay, we'll start again. Here we are. I have to. And then after I do that, I can't find the pointer. It's always the same. Okay, so what happens is this part in the cell twists. It twists, and it opens up, starts to open up these helices. See, when this part in the cell rotates counterclockwise, it's opening up. It's going like this, opening up these helices, and that's looking close. So by the G protein binding, it starts to open up the pore. But we noticed that pore to us looked like it wasn't open all the way. Didn't open all the way. And we also did some other experiments. And the other experiments were, with this protein structure, without the G proteins, we made some point mutations. That means one at a time, altered some amino acids that look to our eyes to be important in the mechanics of the way the protein works. And one of those mutants make the channel open even when you don't have G proteins. And I can show you the experiment that shows that. So here is the same experiment we just did, where you add acetylcholine, the currents turn on, and then you add the blocker and they are inhibited. Here in this mutation, where one amino acid is changed, notice that when you add the potassium to the outside, you have a big current, and when you add acetylcholine, there's very little current more. Now you add the inhibitor, and you notice that a lot of the current that turned on in the absence of acetylcholine is the GERK channel. So what that means is, for some reason, we broke this channel, and it's open all the time. So what does its structure look like? So this is the sort of investigative work you have to do in this business. If you just get one result, you don't have all the answers. You have to keep testing. And so here, I will now do the morph movie from the closed channel to the shape or the architecture of this mutant channel that is always open. And let's see what happens. Notice here there's a bigger movement in a real opening of these helices. And when you look down the center, you'll see that there is both a rotation and a cracking open, a sort of rocking, you can't see it very well, of the subunits here. So it both twists and it cracks, and it rolls like that. And in fact, this kind of movement, when we zoom in, you'll see opens the pore quite wide. So the gate of the ion conduction pathway is opening. Now, this is the mutation that is active even without the G protein. And so, what does that tell us about what the G protein is doing? Well, if you now what I'm going to show you is a different movie. It's a movie of what happens if you morph first from the closed channel to the G protein activated one, then the G protein activated to the mutant. You'll see what happens when you put these together. There's a message. So there's a rotation, then a rocking. And you'll really notice this when we look straight down. So now it goes back, does it twice. This part rotates, then they rock. And so when we look down, you will see that, in fact, the G protein binding causes a rotation, and they begin to open. Then what happens is the subunits rock, and it cracks wide open. So there's a sequential event here, and I'll show you in a little diagram on the next slide. The diagram is here. So what it looks like happens is you have a closed channel. The G protein coupled receptor is activated by acetylcholine. The beta-gamma subunits become free to bind to the potassium channel, and you get an intermediate structure, an intermediate architecture where there's been a rotation in this. It begins to open, but then what happens apparently is there can be a further conformational change that opens it wide. Now, when I saw that, I realized, aha, this makes sense from the electrophysiology or functional recordings I've seen from many labs for many years, and that is if you isolate a patch of membrane with the channel in it and add the G proteins, what you observe is so this is just showing two different voltages, it's current and time. When the channels open, it's as if they don't want to be open. They flicker open, but they don't stay stably open. And so what apparently is happening is the G proteins bind, and it's in a position to open, but then it must jump back and forth here. It's completely consistent with the final opening occurs after the G protein subunits bind. So it's a sequence of events. But imagine what's happening. I mean, when my heart rate slows down, the vagus nerve stimulates, acetylcholine comes on. Acetylcholine is poured onto a receptor, activates the G proteins. The G proteins diffuse over, attach to the membrane, bind to the potassium channel, and then shift it to a conformation that can then flicker open and closed. Why it's set up with this flickering open and closed, we can only keep studying it and wonder what nature is up to here. There are multiple regulating factors in here. I didn't mention what PIP2 is doing with this channel, and sodium ions also do with this channel, but I suspect it has to do with the fact that this channel, not only do G proteins, but multiple stimuli can feed in to tune its degree of openness. Why that slows the heart is because you get more potassium current, and the depolarizing signal, the action potential, of the pacemaker cells in the heart take longer to fire because there's more potassium channels open, forcing the membrane potential to be at the resting potential, the negative potential. The final, just in the last few minutes, I want to talk about a third example, just very briefly. And in a way, in my mind, it's for personal interest, it's been the most interesting example. So there's a different potassium channel in the control. It's not a lipid that controls it. It's not a G protein. It's actually voltage itself. In this kind of control, I'll introduce the following way. Imagine that what I'm showing here is a long tube, a cylinder, and I'm just making a cross-section through the cylinder. So this is inside the cylinder, and this is outside, and this is outside. So this is a very long tubular-shaped cell, an axon, an extension of a neuron, of a brain cell, the kind that lets me do this. Okay. Now, we know that a nervous impulse, when I wiggle my hand, wiggle my fingers, nervous impulse originates, comes down. Now, I'll introduce it this way. If you imagine we were focused on one little part of that nerve, that axon, and it's running here from left to right, and we look right at the part with the action potential, the electrical potential is flipping. Imagine we look right there. What we would see is an open sodium channel, or some sodium channels open. Sodium would be rushing in, making the membrane positive inside relative to the outside. So this whole cylinder becomes positive right in this place. And in that snapshot, we would say that membrane is, compared to everywhere else, it's negative inside because of the potassium channels. Right here, because of the sodium channel, it's positive inside. Now, if we took a snapshot a millisecond later, this positive potential inside would move. If the impulse traveled from left to right, it would move a little further to the right, then a little further. So in a sense, this picture would keep appearing down here, like dominoes. And the reason that happens is because these two, the sodium and the potassium channel in the neighborhood, just to the right, are closed. But now they're going to open. The sodium channel first and the potassium channel second. And they're going to open. Why? How did they know to open? They knew because this channel opened, flipped the polarity, made it positive inside, and it was felt by the ones over here. And then they open. Now, so this is a funny thing I'm describing and a little complicated, because I started out this talk by telling you that because there are ion gradients and channels, that they electrify the membrane. But now I'm telling you that the electricity or the voltage on the membrane is actually telling whether this kind of channel can open or close. It's controlling. So that's recursion. So the channel sets the membrane potential, but the membrane potential sets whether the channel is open, feeds back on itself. Sodium and potassium channels with this property are called voltage dependent. And they're very similar. The difference is sodium channels conduct sodium, potassium channels conduct potassium, but they have the same architectural part that lets them do this, that lets this happen. And if we made a different picture where this now I'm showing as the membrane, so I'm showing the top half of this cylinder, this is outside the cell, and this is inside the cell. And suppose this were a cell at rest. And then I come along here, and I tell that sodium channel right there to open. Or I say, or I stick a pin in the cell right there, so that sodium rushes in, and it goes from its rest negative inside to positive inside. What we would see a millisecond later is it would spread over there, and it would spread over there, and it would travel across the surface of the cell, analogous to what happens if I throw a pebble into a still pond and watch the waves ripple out. And if that is on a cylinder, it goes down the cylinder, okay? And that's the nerve impulse. But it's happening because of a flip of this polarity. And that's happening because these channels open because they feel the membrane becoming positive inside. Why does that happen? The basic idea is this. You can understand it in a cartoon. Imagine you have a channel, just like the ones we've been looking at. Say it's a potassium channel, but in this case, it's going to be a voltage-dependent potassium channel. So what it has is some extra structures on it that we call voltage sensors with a particular architecture that are going to give the channel this property of reading the voltage. And you can see in the cartoon what I've shown here. I'm showing that this part of the protein has some positive charges in it. Say amino acids, certain beads on that string that carry a positive charge, and they're stuck there. Now imagine that when the channel is closed, those positive charges are down here. That means when the membrane's negative inside, it will pull them down and close the channel. Now imagine when the membrane is made positive on the inside by a nearby sodium channel opening. The positive charges will get pushed to the outside, and it will open this. So you see what this is, it's an electromechanical coupling device. If there are any electrical engineers in the audience, this is nature's analogy to something called a field effect transistor. But it's a very, very sharp turn-on field effect transistor. And this is what lets us make action potentials or nervous impulses. Now here is the architecture of a voltage-dependent potassium channel. And what you have, this looks very different than the potassium channels I've been showing you. But I'm sure you're all looking at it saying, where's the potassium channel? And you probably see it inside the middle here. You can recognize that. I'm sure you can all recognize that now and say, when you see one out on the street, you'll say, potassium channel. Okay. Probably after this, you'll never want to hear about a channel again in your life. But there's a potassium channel in there, and that channel is surrounded by 4 voltage sensors, one on each subunit. This part is extending into the cell, and it does some other things. It's important for certain properties of this channel, but it's not essential for the voltage sensing, so we won't talk about it right now. The architecture we're interested in is the part that's crossing the membrane. And so here is a different representation. Now you can more easily see in blue the potassium channel that crosses the membrane. The membrane's running here. You see the potassium ions. These are the voltage sensors. And notice the way they're attached to the pore is very interesting. They're attached by this horizontal structure, an alpha helix structure. There are 4 of them, so they wrap around like a cuff or almost like a purse string around the pore that can, and in fact, it's surrounding that part that we watch open and close in the other kinds of potassium channels. And already we look at that and we're suspicious that, in fact, somehow this thing can probably change its shape and push down and close that. This channel is open, okay? And so what do we see if we now break off the voltage sensor, just to look at it more closely? And so we'll break off the voltage sensor and this helix for reference. And here that is. And so you can see now, here's the horizontal helix that's contacting the pore. And in fact, you see a part I've shown in red that is very important because we know from other experiments that it undergoes a big movement when you change the voltage. These amino acids I've shown here carry a positive charge. This one, this one, this one, this one. And by that, they're electrically plus charged under normal conditions, under normal physiological conditions. So what have we? We have a transmembrane protein with a helix, this structure attached to this horizontal helix that is pushed against the pore, and we have positive charges here. Now imagine this is embedded in the membrane, and the membrane is what gets charged, a voltage across it. And so there's an electric field in the membrane that can work on these charges. When the membrane's negative inside, these positive charges are pulled down. And when it's positive inside, they're pushed up in the position they're in now. And in fact, so that you get this sense that you have a little electromechanical coupling device. Now, the structure here, you know, I said to you it's in a way to me the most interesting, but also the most complicated. The complication is we only have one picture of this in its open conformation. Unlike the others, where we could look at open and close by adding or taking away a lipid PIP2, by adding or taking away the G proteins, in this case, we've not been able to add or take away voltage. You can't do that. You can't titrate it in and out. You have to do other tricks that we still haven't solved the problem. But you can already see here in it the principle of how this is going to work, because this positively charged unit can move this lever and close the pore this way. I say this is hypothetical, but this is based on the closed pore structure of other potassium channels. And presumably these little levers push down and cause the pore to close. And so it allows, in a sense, though the voltage sensor is a charged protein that does just this, shown in this structure, undergoes a conformational change. And so let me end, and so in a sense it's this process that we still work on with intensity that allows a channel in the neighborhood of a voltage change to read that voltage change in itself open, which is essential for the propagation of the nerve impulse. Let me just end by saying there's much work to be done in understanding. There are controls on different potassium channels that I didn't even mention here. There are ones, for example, that open when you touch the cell. So I've talked about G proteins activating channels and lipids and voltage, but there are actually ones that open when you touch. And we don't understand yet that sense in neurobiology, in neuroscience. We know how vision works, we know how, in principle, the initial steps of smell and taste, but we don't know touch and hearing. There are channels that respond when you touch, and there are potassium channels that respond when you touch. How does that work? We don't know. We actually have the structure of one of those, and we look at it, and it gives us hints, but we don't quite know yet. There are potassium channels that respond to other elements like calcium, and it's an incredible diversity, and we're just at a level of trying to understand the biophysics based on the architecture, but I think someday what we're learning will help us to be able to control the way channels open and close with molecules that hopefully will help in certain disease states, because the activity of potassium channels underlies everything from nervous impulse propagation to fluid secretion, blood pressure control, hormone release, such as insulin. If we can understand the architecture and the chemistry at a deep level, I believe we can regulate those processes to benefit. But we're still at a very early stage. And I want to end finally with a very important slide, because all of this work is actually done not just by me, but by a group of scientists who work with me over the years. They come from all around the world, from Europe, Asia, South America, the entire world. And in fact, it's the greatest part of being a scientist. And every time I ever complain anything about my job, like I didn't get my grant, I always think, what a lucky person I am, because actually, you know, people from all around the world come just to work with me and learn from me and think with me and figure out these problems. This is a list of people relevant to the problems I talked about today. The PIP2 activated channel, the work was carried out by Xiao Tao and Scott Hansen. Scott is now at Scripps Institute in Florida. The G protein gated channel work carried out by Matt Wharton. He's beginning to look for a job now. So I would try, he's very good. The voltage-dependent channel by Steve Long, who is a professor at the Memorial Sloan Kettering now, also Xiao Tao, and Ernest Campbell, who's worked with me for many, many years. That was the work I talked about today. Thank you very much.
Bien. J'aimerais juste vous rappeler, si vous avez des questions, de remplir les fiches. Les hôtesses vont passer les prendre maintenant. Alors, nous avons un petit problème parce que toutes les questions sont très bonnes. Merci beaucoup. J'espère que vous n'avez rien prévu avant 2 heures du matin.
I will. Okay. These are all fantastic questions. I'll take them at random. How does a snake or spider venom block the channel? Do you want to translate? No.
Vous avez traduit? Est-ce que vous entendez la traduction? Je vais traduire. Comment le venin du serpent ou de l'araignée bloque-t-il le pompe ou les canaux?
Oh, the pump of the channel. Okay. Wonderful question. So we know a lot about how, in the case of potassium channels, and it's interesting that the person asking the question asked snake or spider. They do it differently. So snakes, well, it depends on which one, but snakes, the ones we know most about are the mamba snakes from Africa, the green and black mamba, very deadly. The recommended treatment if you're out in the wilderness and you're bitten by one and you're very far from medical attention is lie down under a tree and enjoy your last few minutes. So the way it works is it makes the venom has a protein, a small protein. By small, it would fit just on the top of the channel like a plug. It's about 35 to 40 amino acids or beads on a string, folded up, and it's actually a disulfide bridge to form a little rigid structure with just the right shape to... That's it. Scorpions do the similar thing with a different structure, but it also fits very well into many potassium channels. The snakes, the spiders, though, and the ones we know the most about are tarantula spiders, the very large, hairy spiders. They feature in movies to frighten you. They make a protein that binds to the voltage sensor. So it's very nice. You can, in fact, tell which parts of the channels or pumps are the most important just by asking, how did nature subvert or attack it? And so you have cases where there are plugs that keep the ions from going through, like the plug in a bathtub drain keeps the water from going through, or you have toxins that bind to the voltage sensor and lock it so it can't move and prevent the channel from its normal opening and closing. So there are examples of both. Another question. Do we know of any other systems in nature that imitate ion channels, meaning that the charge transport happens in a similar fashion? The answer is yes. It turns out that there are many bacterial toxins that work. So talk about imitation. They work by making channels in membranes, but they're not nice channels like a potassium channel. They are non-selective. So they go into the membrane and make a pretty big hole, but it is a channel. And when that happens, then, of course, the way I describe the cell as having a system where it electrifies by the membrane, by having selective channels. Imagine what happens if you now insert non-selective channels into the cell membrane. Now the potassium runs out, the sodium runs in, the whole thing collapses, the gradient collapses, the cell dies. And that's why it's a bad toxin. There's one toxin known very well for pumps. The details of how it works isn't yet known, but it's a very famous toxin called palitoxin, and it's from the deadly coral toxin. And this toxin, when it binds to the sodium-potassium ATPase pump, it holds it open, completely open, and makes sodium and potassium rush, so it no longer pumps, and it collapses again, collapses the gradient, and kills the cells. It's one of the most deadly toxin in terms of the amount of death it could cause for nanogram of material. So paletoxin is very deadly, and it basically locks the pump wide open to make the sodium run in and the potassium run out. Yeah. So in a sense, these are mimics of channels in nature that actually are used to subvert the natural system to kill the cell. Yeah. Okay, genesis of the potassium channel. When did they appear and in what living creature? So we can find potassium channels throughout the tree of life, in the bacteria, the archaea, in the eukarya, fungi, plant, subsets of eukarya. So they're completely throughout. You don't find all the same potassium channels in us as in all of the organisms, but they all have the same pore. So all we can conclude is that the potassium channel arose very, very early in life. And in fact, the pore is essentially invariant. We say it just ignored the prokaryotic-eukaryotic boundary. So apparently that solution that evolved very early on in life was maintained throughout the tree of life. Oh, why does the signal, the electrical impulse, go in one direction along the axon, left to right, and not in the other or both? Good question. So it turns out if you go to the nerve axon and start, so the way I described it is I said, suppose the signal is coming from left to right. It will continue to the right. And the reason I worded it that way is because if you suppose you take an axon that has no impulse, and now you start an impulse in the middle of the axon by sticking a little pin in it, for example, letting sodium rush in and making it positive, it will go in both directions. It will go in both directions. So then that raises the question, well, why, when the impulse is going from left to right, doesn't it keep spitting off impulses in the back direction, right? And the reason it doesn't, and again, it's sort of the clever beauty of nature, those channels, when they open, they then inactivate, which means just after they've been open, they can't be triggered again for a little while. We say they're refractory. And it has to do, in the way we don't understand completely all the architectural changes that take place, but an activated channel isn't yet primed to go for several milliseconds. And this is a very important aspect. What must be happening is the channel opens, undergoes a conformational change, and before it gets back to its closed prime state, it takes a little while. It has to go through some different shapes. But that's important because if it immediately could cause an impulse, every time an action potential would go back, it would fire action potentials again in the backwards direction, and the whole nervous system wouldn't work. I mean, you'd have one impulse, and then you'd have a seizure, and it would be over. And so it's because the channels go into a refractory state just after they've been activated. And kinetically, it takes them a little while to prime again. So a potassium channel, how do you detect the closing and opening? And how fast is this process? Well, you detect it, one of the ways you detect it is by the methods I showed. You can actually watch it open and close when you're looking at single channels. And so the process is typically milliseconds, tens of 100 microseconds to millisecond, which on molecular time scale is fairly slow. It's about as fast as it can happen for a complex movement of a protein. And our recording devices can go that fast. So we can actually keep up and measure the kinetics of channel gating. You assume that the structure and shape of the protein is the same in the crystal and within the cell. Do you have evidence for that? And so in every case, we always look at it and wonder if it is. And so it's a very good question. So that's why it's important to carry out experiments on the channels with this in the cell membrane to make sure what you see makes sense with what you see in the crystal makes sense with what you observe in the membrane. And let me tell you an example of a case where we looked at it and we could tell that it probably wasn't in a normal conformation. And in the membrane, we took the channel and put chemical reactive groups on part of the protein to test whether the shape of the protein, the part that sticks out of the membrane on the outside or the inside, is in the same place we see in the crystal. And by doing that, you can test to make sure everything adds up for the channels in the membrane and what you see in the crystal. It turns out for most of the time, for most proteins, their shape is or the few shapes they can adopt are the ones they like to adopt. They're energetically more stable than unnatural states. So protein crystallographers are very lucky for that. So in almost all of the time, and you could say, well, how do you know? Well, it's by experiments that just all kinds of functional experiments that you do to make sure everything adds up. This group seems to be close to this group according to this certain functional experiment. Is it close in the crystal structure, for example? So there are many ways of looking at the protein to try to make sure everything adds up and there are no big discrepancies or even small discrepancies. So that's what you have to do. Is one of the components of regulating the potassium channel used in medicine for people that have a problem with heart rate? And do we know how does the potassium channel make the heart rate go slow? Yes. So the second one is easy to answer, and I didn't explain it very well, but it turns out there are pacemaker cells in the heart that tend to go at a certain frequency, but that frequency can be modulated. And the way the pacemaker cell works is if you put an electrode in it, measure the membrane voltage, you would follow a negative voltage, like I showed you, it would slowly become more positive, and once it reaches a certain point, boom, action potential would come back. And with the action potential, a sodium channel is open, cause the depolarization, then more potassium channels, voltage-dependent potassium channels open, and drive it back, becomes very negative, and then after a little while, it slowly becomes more positive again, and that has to do with a certain kind of potassium channel that has a gating property that I didn't talk about. But it turns out if you open more potassium channels at rest, you make it more negative. And so the membrane voltage that is slowly becoming more positive doesn't reach its threshold that opens the voltage-dependent sodium channel. So that's why regulating the amount of active potassium channel in the membrane spaces the interval between action potentials. So that was there's a lot of complicated kinetics in there, and it was a sort of a qualitative description of what's happening. But that's it. So that's why stimulating the potassium channel with the G proteins opens more potassium channels, makes the membrane more negative, so the slow depolarization takes longer to reach what's called the threshold potential, to fire the next action potential. Then the question, is one of the components used as medicine to help people with heart rate problems? Not yet, but the pharmaceutical industry would very much like inhibitors of the G protein gated channel I talked about that are specific. And the reason they would like that is because there's an abnormal heart rate problem called atrial fibrillation, and I can guarantee that at least some people in the room have that. And there's, if you're very young and you have it, there are some good treatments due to what's called an ablation procedure, where parts of the conduction system can essentially be burned out to make atrial fibrillation go away. I should mention that half my family has it, and I will have it because genetically, my family's susceptible to it. In animal models, mouse models of atrial fibrillation, where you induce the atrial fibrillation, it's been discovered that if you remove this potassium channel, the heart G protein gated potassium channel, you can't induce atrial fibrillation. So that's why the pharmaceutical industry wants an inhibitor for that. But nobody's been able to discover a good small molecule that has the specificity for only that potassium channel and not others. But I think there are ways, now that we're learning much more about the channels, there are ways of doing that, and that's one of my hopes. I can't, can you measure? I can't read this one. It was a translated one, but I can't read it. So what?
How do you measure the low tensions then? How do you measure the very low voltages?
How do you measure, the tension, okay, the word, voltage. How do you measure the voltages? With an electrode in the cell, and you can easily measure voltages with normal fluctuations of voltages within in a cell membrane, or you can measure the voltage more accurately than the normal variations that occur. And with many experiments, in fact, we use a very fancy electronic circuit where we put electrodes in the cell, and the electronic circuit actually controls the voltage. We put the voltage at what we want it, and we measure the current through the cell membrane when we control the voltage. So, in fact, measuring electrical current and voltages is a very precise thing, and you can do it very well. It's one of the nice aspects of studying ion channel proteins. One question is, my work, am I inspired by quantum physics? Well, yes, I'm inspired by quantum physics, mystified by quantum physics, but not because of my work, because actually proteins and the atoms that make up proteins are pretty heavy. Potassium ions are pretty heavy, and most of what we observe and everything I talked about can be described in terms of classical physics. So quantum physics, in terms of understanding diffusion processes, both ion diffusion and diffusion of a protein from one I say diffusion of a protein, the wiggling and jiggling of a protein from one conformation to the next is mostly classical considerations. Of course, what's not classical are the atomic force, understanding the atomic force fields that are underlying that basically go into understanding those movements of the protein. So that if you want to have a description of the force field to describe the motion in a classical sense, those force fields arise for quantum mechanical reasons, but we don't have to explicitly deal with that. Yet, that said, one of the real problems, if we understood proteins as well as we would like to, in a sense, why wouldn't we be able to take an unfolded polypeptide and put it down and watch it fold and calculate how it will fold up and what shape it would take and what function it would have and how it would go? We should, in principle, be able to compute this. But we can't do that yet. And one of the problems, I'm sure, is at the level of a fundamental understanding of the force fields in proteins. Well, I'll just finish these two unless you're tired of hearing my voice. What happens in the body when there is a shortage of potassium. Would you like to answer that? When there's a shortage of potassium, it's a very good question. So when I was a medical doctor, one of the, if people, for example, with conditions like high blood pressure or other heart problems, something called, it's a bad name, but called heart failure. It's not really heart failure. Are on diuretics, which are medicines that make, so you tend to retain too much water, too much fluid, and they're medicines that make you excrete water. They make you, well, in America we say make you pee. So you get rid of extra water. And the problem with those medicines is one of the things you also get rid of is potassium. And so whenever somebody with high blood pressure and some problems or other heart problems come into the emergency room, we would immediately measure their serum potassium levels. And the reason is because you're on a diuretic, one of these medicines that make you lose fluid and potassium. The potassium level gets low, and that's dangerous because, and think about why. I just described that the voltage or the degree of electrification of all your cell membranes is dictated by the gradient of potassium. And I said there's a high potassium inside and low out. And it's low but not zero. It's around 4 millimolar. And it turns out the voltage is related to a constant times the logarithm of the ratio of the potassium concentration inside to outside. And that outside number is small, so small changes in it have a big effect. And so the body normally will strictly keep your potassium level between, say, 3.5 and 5 millimolar. But once you start to come out of that range, and especially if you come out of it on the low end, you affect the electrical excitability of cells, and that is, you affect things like the heart rhythm. And that can cause arrhythmias by having the low potassium. So the shortage is very bad, yeah. And that's why, naturally, our kidneys strictly control its level, as you'd imagine. The structure of the potassium channel was a long unsolved question. Now that it is solved, what structure would you like to see solved next? Well, you know, in my heart, I'm more of a physiologist than a structural biologist. And many people ask me, you know, what other channels do you work on? And it turns out I do work on a few others, calcium and sodium channels, for example, and chloride channels. But mostly I work on potassium channels because I'm very interested in these control mechanisms. You know, there are 78 on that family tree. And it's not that I want to solve the structure of all 78. I actually don't. But I do want to understand some fundamental mechanisms. I really want to understand this nervous impulse voltage sensing mechanism. And as I might have hinted to you at the end of my talk, we know nothing, very little about touch sensitivity. And I'm very interested in ion channels that can sense this. We don't know the basic physics of that at all. So these complete unknowns are interesting to me. So that's the way I think about it. And then there's the question, are there pharmaceutical possibilities for the potassium channel? And yes, there are. And I think I already mentioned a few of them. And finally, here is a question that we can probably all address. And it's, what could European institutions learn from Rockefeller HHMI? So I'm at Rockefeller University, and my research is supported by the Howard Hughes Medical Institute, and I do consider myself one of the luckiest scientists in America because, and I'll just tell you what I like about Rockefeller and what's nice about funding from HHMI, and then others can make comments. Rockefeller is a small university where we don't have a lot of, we don't have administrative things to deal with. You know, we have no departments. We have a president and labs. We have no centers, nothing like this. We have a few center names that are actually for I'll just say it, fundraising purposes. But at Rockefeller University, we have our individual labs, and we concentrate on the science. And there's not a lot of administrative things pulling us away from the science. It's a great place to think about your work. In the Howard Hughes Medical Institute, I'm very fortunate because it's what I call progress report science. So the way they evaluate, so every 5 years, I come up for renewal. And I will not be renewed if my science is not good. It doesn't matter if you have all kinds of prizes. What they want to know is, what have you done recently? Show us what you did in the 5 years since we last renewed you. And they don't want to know, show us your 35 papers or something. No. They say, show us your 5 favorite papers. That's all. And come tell us about this. And then a little bit, tell us what you're thinking about for the future. So basically, it's common sense. They're looking at your immediate past to project to your immediate future, to extrapolate, to see if you're worth giving another 5 years of support. It's fabulous. It's not a lot of paperwork. I fill out 5 pages of a description. That's it. And hand in 5 papers. And then I sit in front of a panel of excellent scientists who are selected, who decide whether I'm worth funding for the next 5 years. And I think it's great. And I think that when I hope I keep doing work, that they'll keep deciding, but I hope if I don't, they say no. Well, actually, if I don't, I hope I say no before I go in front of them. But that's what I can say about my situation. And probably there'll be opinions about funding and science in comparison to this. There are probably similarities.
So, as an American who moved to England, I can say that Europe is not monolithic, okay? And there are countries like Britain and Switzerland which excel in science, or on a per capita basis are actually, quite comparable to the United States. And there are other European countries that are not doing so well. And I think the countries that do well have Rod's recipe, which is their meritocracies, and where they judge people based on their track record and the prospects for success, and don't pay too much attention to whether they're very important people or have a lot of stature or things like that. The other thing, I think there are 2 other things. One is that the countries that are successful allow group leaders or young faculty members to have independence at a very early age, okay? Whereas the countries that are not so successful, you sort of join somebody else's umbrella organization and have to slowly work your way up to the top. And by the time you have that real freedom, you're maybe in your mid-50s, okay? And I think it's just true that people are more creative and more adventurous when they're younger. Maybe this is not a very popular thing to say, but you can take almost any great discovery, or in fact, you can take any great book that was written in literature where, age should count for experience and so on. And you'll find that people did their best work rather early on. They may have followed it up with important work that sort of built on their breakthrough, but their breakthroughs came, relatively young in life. And so I think that's another thing. The system should foster independence at an early age and give people freedom.
Andrew Holmes, I think I can add to that to say that we're living in a world where society depends fairly quick results for what we do, and that's an increasing pressure. And it is a much greater focus on the translation of discovery to application. And I think what we could learn from what we've just heard is I will quote 2 former presidents of the Royal Society who have said, what is pure science today will be applied science tomorrow, and let's invest in the pure science. It's perhaps not a perfect solution, but it's actually very important.
Yeah, and I'll just follow up on that by saying that, I said I'm very fortunate to be at Rockefeller and to be funded by Howard Hughes Medical Institute. But there are, I am pretty vocal about changes in funding in the United States that are not good. And in fact, there's more and more emphasis on quick results. And what, there's a term in biology now called translational research. It's a term that I really worry about. We have an NIH director who's just turned our National Center for Research, ended one very important aspect of NIH and started a Center for Translation or some name like that. This worries me, actually, because I really firmly believe, what you just heard, and that is that fundamental work is what gives rise to application tomorrow.
Actually, that's a worry. Europe used to be known for, making really breakthrough fundamental discoveries that often took decades. And I could, just to give examples from Rod's talk, the whole idea of action potentials, came out in Cambridge. The idea of how to solve protein structures, was also done in Cambridge. And the idea of how to get crystals of proteins that were normally embedded in membranes came out of Germany. So it's had a long track record of doing very fundamental work. But I see increasingly European governments, and it's especially top-down, often from the EU or things like that, but as individual governments as well, constantly saying, we want to do more translational research so that we can be more like America. What they mean is that they want to be like America in terms of converting basic discoveries to important applications. They see Genentech or other companies like that succeeding, and they want that. But that doesn't come from funding translational research at universities. The thing that makes America particularly strong in this respect is this idea of venture capitalism and people willing to take a risk to start companies. So it's a psychological mindset that makes America very good for starting up companies and so on. It's not because, American governments fund translational research and that's what people should do here, okay? And risk-taking is a cultural thing. In order to be able to take risks, you have to have a society that doesn't mind you failing, so that when you start a company and you fail, you're not regarded as a failure. Rather, you're regarded as a courageous person for having taken the risk and started a company. That's the difference between what I see as a more American culture and some of the cultures in Europe where if you fail, it's considered a terrible thing.
Ok, merci beaucoup pour la réponse à toutes les questions, pour les questions, pour la discussion. Et je vous donne rendez-vous demain à 18h30 pour les accomatériaux. Merci, bonne soirée.